Hi, in this video we will be discussing about the mechanism of action of penicillins. Penicillins are one of the 4 beta-lactam antibiotics. These are bactericidal drugs, and it works by interfering with bacterial cell wall synthesis. So today we will be discussing about the structure of bacterial cell wall, and then we will go through the process of bacterial cell wall synthesis. Here we are looking at the bacterial and human cell. As you can see, the cell wall is unique to a bacterial cell, whereas human cells do not have cell wall. It only has a cytoplasmic or cell membrane. Cell wall is a layer that completely surrounds cytoplasmic membrane, and it protects the cell from mechanical disruption and osmotic pressure damage. This is a simple diagram showing cell wall of a gram-positive and a gram-negative bacteria. So this is the cell wall, this is the cytoplasmic membrane. The cell wall is mainly composed of peptidoglycan, and as you can see, the gram-positive bacteria, they have a thick peptidoglycan layer as compared to gram-negative bacteria, which has a thin peptidoglycan layer. The gram-negative bacteria also has an extra outer membrane, which is usually absent in the gram-positive. This peptidoglycan layer is an important and a unique component of a bacterial cell wall. Peptidoglycan is a polymer of polysaccharides and peptide chains. Polysaccharides contain an alternating N-acetylglucosamine and N-acetylmuramic acid. These are attached through glycosidic bonds. Now let's look at the peptide chains. These have 5 amino acids with 2 molecules of D-alanine. It also has a pentaglycine side chain attached to the 3rd amino acid. This peptide chain is linked to the N-acetylmuramic acid. Now this represents a monomer of peptidoglycan. The peptidoglycan precursors are synthesized in the cytoplasm and are transported across the cell membrane for assembly into linear polymers. These linear polymers must be further cross-linked by transpeptidation reaction. Cross-linking provides stability and rigidity to the cell wall. In this reaction, a glycine of one strand that is located in the 3rd amino acid forms a peptide bond with the D-alanine in the position 4 of another strand. And then it removes a terminal D-alanine from the peptide chain. This reaction occurs in presence of an enzyme called transpeptidase, which comes under the group of penicillin binding protein. This transpeptidation reaction is an important step for cross-linking of these polymers. Penicillins are structural analogues of D-alanine, hence it binds to these penicillin binding proteins. Thus, it inhibits the enzymatic activity of transpeptidase, thus inhibiting bacterial cell wall synthesis, and ultimately the cell dies, likely due to the mechanical disruption and osmotic damage. A few important points to note is that the cell wall is synthesized during bacterial replication. Hence, penicillins are more active against rapidly dividing bacteria than those in the resting or stationary phase. As mentioned earlier, human cells have no cell wall, so this inhibition of bacterial cell wall synthesis has no effect on host cells. And finally, as discussed earlier, the gram-negative bacteria has a single layer of peptidoglycan, whereas gram-positive bacteria can have up to 40 layers of peptidoglycan. And this may be the reason for higher susceptibility of gram-positive bacteria to penicillins.